Hi, this is Cells Part 2, the HL Origins of Cells. Starting with endosymbiosis. Eukaryotic cells are believed to have evolved via endosymbiosis, where one cell engulfs another cell, and that cell became assimilated into that original cell's cellular structure. Elaborating on this, current evidence suggests that all eukaryotes evolved from a common unicellular prokaryote that had a nucleus and reproduced sexually. This prokaryotic ancestor at some point engulfed another aerobic bacterium, which over time lost its independent utility and developed into what today is known as the mitochondria. And a subsequent internalization of a photosynthetic cyanobacterium likely contributed to the development of the chloroplast in plant cells. A diagram that could explain this would be this one right here. If I can get it, it displays the original prokaryote here, then the membrane infolding as here, and then other bacteria being absorbed and later made into part of itself. That is endosymbiosis. Now, evidence for endosymbiosis. Both mitochondria and chloroplasts are organelles that are suggested to have arisen via endosymbiosis, and these organelles are therefore expected to share some characteristics with bacteria as a consequence of their origins. Evidence that supports the extracellular origins of these organelles include their membrane. They both have double membranes. The outer membrane may have been vesicular in origin, but it's still a membrane. Antibiotics. Both organelles display susceptibility to certain antibiotics, such as antibiotics target prokaryotic features. DNA. Both mitochondria and chloroplast possess their own DNA, which is naked and circular, like in prokaryotes. And division, as well as ribosomes. Division. Both organelles divide by a process similar to binary fission, which is a common reproduction method among bacteria. And ribosomes. Both of them have 70S ribosomes, such as just like in prokaryotic cells. Now, that's it for endosymbiosis and the origin of some eukaryotic cells. Moving on to multicellularity. Multicellular organisms are composed of multiple cells, operating in unison to support the total life forms. All plant and animal species are multicellular, along with most fungi and several species of eukaryotic algae. Multicellularity has evolved repeatedly and offers several distinct survival advantages. Being multicellular allows an organism to exceed size limits normally imposed on by the size to volume ratio limitations for a single cell. Multicellular organisms can have longer lifespans as the organism can survive the death of an individual cell. Multicellularity fosters complexity by allowing the differentiation of cell types within single organisms, so different cells can do different jobs. And one challenge faced by multicellular organisms is the occurrence of cancer, whereby cells fail to regulate their normal pattern of growth within the organism. Now, multicellular organisms are capable of completing functions that unicellular organisms could not undertake. This is due to the collective actions of individual cells combining to create new synergistic effects. These new functions are called emergent properties and arise as a consequence of cell specialization. In a multicellular organism, the different expressions of genes causes cells to become specialized and develop unique functions. The differing patterns of gene expression may be coordinated by extracellular signals or triggered by changes in the environment. In multicellular organisms, cells of the same type may be grouped together to form tissues. The functional grouping of multiple tissues results in the formation of organs. Organs may interact to form organ systems capable of carrying out specific body functions, and organ systems collectively carry out the life functions of the complete organism. So that's multicellularity in a nutshell. Some examples, beside specific plants and animals, would be like a dog, a cat. All of them have unique body functions due to their multicellularity and cell specializations, since specific cells can do specific things. Moving on to organic material. All cells and organisms are constructed from a complex carbon-based macromolecule, or a set of them, called organic compounds. Organic compounds can vary greatly in structure and diversity due to the chemical properties of carbon, which can form 4 covalent bonds. The generation of organic compounds from inorganic sources is considered to be a fundamental requirement in the initial formation of cells. The large variety of organic compounds allows cells to undertake a greater range of chemical processes needed to sustain life. The spontaneous formation of simple organic compounds required specific prebiotic conditions. A lack of free oxygen in the atmosphere meant there was no ozone, which resulted in higher levels of ultraviolet light penetration. Higher atmospheric concentrations of carbon dioxide and methane resulted in significantly higher temperatures. These conditions no longer exist on Earth, and hence the chemical processes required for carbon compounds to form spontaneously do not now occur. Now, the Miller-Urey experiment recreated the postulated conditions of prebiotic Earth in order to demonstrate the non-living synthesis of organic materials. In his experiment, water was boiled to vapor to reflect the high temperatures common to Earth's original conditions. The vapor was mixed with a variety of gases, including H2, CH4, and H3, in order to create a reducing atmosphere, which is just an atmosphere with no oxygen. This mixture was then exposed to an electrical discharge, simulating the effects of lightning as an energy source for reactions. The mixture was then allowed to cool, concentrating components, and left for a period of about a week. After this time, the condensed mixture was analyzed and found to contain traces of simple organic molecules. Now, that's organic material. Finally, moving on to abiogenesis and the functions of life. All living organisms are self-sustaining, meaning they are able to maintain the conditions needed to continue their existence. These conditions include metabolism, reproduction, sensitivity, homeostasis, excretion, nutrition, growth, and movement. All of these things are what it means to be alive. They're conditions that a living being should be able to replicate no matter what, and that's what makes them alive. Now, cells are highly complex structures that can currently only be produced via the division of pre-existing cells. However, the first cells must have spontaneously arisen from non-living material, which is known as abiogenesis, which I mentioned earlier in part 1, as well as the processes to life. The process is theorized to have occurred over 4 key stages. 1, catalysis, where simple organic molecules were synthesized from primordial inorganic molecules. Self-assembly, where more complex polymers were constructed from these simple organic molecules. Self-replication, where certain polymers formed the capacity to be duplicated, which is what enables inheritance. And compartmentalization. These molecules become packaged into membranes with unique internal chemistry. The theory of abiogenesis has been difficult for scientists to test for a number of reasons. One being the exact conditions on a prebiotic Earth no longer exist, and cannot be replicated under controlled settings. The first protocells did not form fossils, so there's limited evidence on which to base hypotheses. Alright, now that is all of the HL content. And that concludes cells part 2. Thank you.